So here is there is a selfing. It was the F1. So again on selfing, what will you find? You will find the 4 types of the progenies. You will be getting TT, right? TT and small t. Here it is very important to let you know what exactly it is showing. The zygotic condition, the zygote is formed or the baby is formed for that particular character, and that the character was what? Character was the height. So what do you see? This plant is still tall, okay, but this plant is also tall and this is also tall. Why? Because T allele is dominant over the small t, and here also is capital T is dominant over this smaller allele, and this allele, these are the homozygous recessive type, and they are, that is why this plant group is what? This is a dwarf one, and all the 3 were what? They were tall. Homozygous dominant or the in heterozygous condition, they are all the tall. So as I had told you, the alleles which are dominant in just homozygous condition, right? The alleles which are dominant only are alleles which are expressed only in homozygous condition are known as recessive allele. But the alleles which are expressed in both the condition, heterozygous and homozygous, those alleles are known as this. So capital T, capital is the dominant allele. So it is the dominating. In one of the experiment what the Mendel had conducted, actually there was 900 around tall plant was obtained, and another category 299 were a dwarf plant. It was not exactly, it is a ratio, actually it is a ratio, it is a 3:1, it is the ratio, but exactly Mendel had got around this much of the value. This was you can compare, it was 3:1 near about ratio. So that is being justified. If a monohabit cross is conducted, then in F1 and Fn we get the only one type of the organism, that will be which of the trait is dominant. In subsequent F2 generation, what happens? In subsequent generation, what happens? We get the organism in the ratio of the 3:1. 3:1 basically here we will be telling you what is the phenotypic or genotypic. But let us see. Actually Mendel had got this one ratio, this type of ratio he had got, and he summed it, he made it what? He gave the proportion of these values into 3:1. Exactly, it was not 3:1. It was a 900 something. One was 1,000, another was 300, something like that. So don't take it was as accurate as 3:1. No. It was near about the value to facilitate the study, to make the study easier. This was the Mendel concept. So here he has the use certain. We use the term or we find that there is a phenotype or the genotype ratio. Phenotype. Look at phenotype and genotype. As you guys have already heard the name of these two terms. Phenotype, as you see the organism from its outward appearance, what I am looking, you guys, as I am looking, this is my phenotype, but the internal genetic makeup is its genotype, right? Generally when we conduct an experiment, and inferences is to be drawn, then we go on to the phenotype, not onto the genotype. Genotype is always to ascertain what will be the type of the organism. Is it really homozygous or heterozygous? Then we conduct the different type of cross to ensure the organism is what. So in general, the prediction comes from the phenotypic assessment. So phenotype is what? It is the outward appearance of the organism. It is the outward appearance. बाहर से कैसा दिखाई देता है, ये वो है. And this is it is the genetic makeup of genotype is the genetic makeup of organism. So it is generally not easy to get what is the genotype the organism, but in transmission genetics we can easily calculate, we can easily predict what is the genotype and what is the phenotype. So look at here, as I told you a few minutes back, we conducted a cross, right? So we get the T, those were the phenotype. This is the tall and this is the tall, this is the tall, but this was the dwarf. So phenotypically dwarf, But if you look very carefully, this is the heterozygous condition, and this is also the heterozygous condition. So these two are what? Yes. So what you see, the genotype is different. Phenotype is same. These are all the three are the tall. Is that clear? But actually, the two are heterozygous condition. One is this pure, or you can say homozygous dominant. Another one is the homozygous recessive, or this is the. So what you see, the genotypic ratio is basically 1:2:1, but the phenotypic ratio is the 3:1. So what do you see? Yes, of course. What you see is that in genotype, I beg your pardon. So in genotype, what you see, the organism has to be ascertained whether it is in heterozygous condition or it is in homozygous condition. But in phenotype, we can predict. We can predict organism is actually tall because it is the outward appearance. We can easily say that this is the phenotype and this is the genotype, but it is the phenotype on the basis of which we can predict the organism is what is the type of the organism. It is easiest way of assessment, but to ensure the genotype, we go for the calculation, we point it out. But and again you can see these are the different, this is the tall and this is also tall, this is also tall, but there is a difference between the three. How? These are the in heterozygous condition, as I gave you a few notes back. These are in heterozygous condition, this is in homozygous dominant condition, and this is in homozygous recessive condition. So this was so pure. So this is the pure, these are the pure. Sometimes we also mate with a certain term, true breeding lines. Actually, when Mendel conducted the experiment, taking the Pisum sativum as a model plant, then he used the term true breeding, tall and true breeding dwarf are the pure line. We generally use the true breeding tall. A few minutes back, same work is I am dealing with the true breeding tall and true breeding dwarf. So it is nothing actually these are actually in homozygous condition. Is the TT, as you have already seen, TT and TT. So homozygous recessive, homozygous dominant. Okay, so these are the pure line, or these are the true breeding lines. Why? Whenever you use whenever you self-pollinate them, then what you get? You will get every time you will be getting the same type of organism. You will be getting the same type of organism. This is the character of the true breeding lines. Similar will happen with true breeding dwarfs. So is that clear? So let us move ahead further. Suppose you conducted a monohybrid cross, taking one character into the account, and in F2 generation or F3 generation, whatever kind of proportion of the organism you get, ultimately you have to check that my organism is homozygous or recessive, homozygous recessive or homozygous dominant, or what is the nature of. To ensure that, we generally conduct a test cross. It is a very important cross. It is a test cross. So what is this test cross? So test cross, basically this cross is nothing at all. This cross is just to ensure that whatever organism we have is homozygous or heterozygous. For example, we take, suppose we have this is my F1, right? And you have to confirm that what is the nature of this one, it is homozygous or heterozygous. Then immediately you will be crossing with the crossing it with the homozygous recessive, or you can say you will be crossing with the one of the recessive parent. And then ultimately, whenever you conduct this type of cross, then you will be getting the certain ratio of the organism. On that basis, you can predict that this F1 is a heterozygous or homozygous. So let us see, if you conduct this one, so and this will forming two type of the gametes, right? And this is forming just one type of. So we will be getting the TT and TT, right? So if the organism, we are getting the 1:1 ratio. Contrary to this, if the F1 organism is the homozygous dominant, suppose it was something like this, and we are again conducting test cross. So we are selfing with the or pollinating with the what, with the homozygous recessive, then what will we get? We will be getting just one type of organism. All are tall, and here tall and dwarf. We are getting the two types of. So what do you see? Yes, we see that if the organism to which we are conducting test cross is in heterozygous condition, then we will be getting 1:1 ratio. If on the other hand, if the organism we have is in homozygous condition and we are conducting the test cross, then and we are getting just what, only one type of plant, it means this my organism of choice was what, it was simply homozygous dominant, or simply you can say it was homozygous condition. So this is the concept. Now I will talk a certain few concept of how the cross is made, how to make the things very easy. Suppose you I just talked about the monohybrid cross, right? I just talked about the monohybrid cross, and in monohybrid cross I calculated we can generally suppose monohybrid cross is there and we are getting the ratio of 3:1, as you have seen a few minutes back. 3:1. How when you conducted the what, selfing, you conducted the selfing, you have seen just then what you get? You get this right, this and this. Now before dealing with all these things, let us go into the aspect of statistics. What exactly is mathematics is involved in framing the gametes or in creation of the zygote, right? So as you told a few minutes back, this is the 3:2, but actually what is the way of the gamete formation, or what can be different way of combining these gametes, and how the allelic frequency can be arranged. So to calculate that, there is a statistics. One is that is the statistical approach involved in it, and this is nothing but it is a product rule. It is a product rule. Yeah, you may ask, what is the product rule? Yes, I will tell you what is the product rule. In these events there is a product rule and addition rule involved. Addition rule involved. What is the product rule? These are the probability rule. It simply says that if there are two independent events, if there are two independent events, right? Independent events occurring simultaneously, occurring together, you can say occurring together, then the total probability is the product of their individual probability. That is the product rule. Then total probability is product of their individual probability. Okay, suppose what is the probability that in a family the first baby will be girl and second baby will also be girl. So let us look. So what are the two events? Are in a row, two events. So the probability of the first baby will be girl is what 1/2, and second first baby will be girl is the 1/2, and second baby is also 1/2 because it may be a boy. Similarly, that is why I am using 1/2 and 1/2. Then what will be total probability? Total probability will be 1/2 into 1/2 is equal to 1/4, and this is the product rule. And what is the addition rule? Addition rule says that it may also apply to the product rule situations where actually there are two events. Here the two events occurring independently. In case of addition rule, there are two events which are exclusive to each other. Suppose we have two dice and we thrown together them. What is the probability of getting 1 or getting 6 in both of them? Again, the probability of the getting 6 is 1 by 6, right? And probability of getting 6 in another dice is 1 by 6. So what will you do? Again, the product rule is applies, and you will be putting 1 by 6 into 1 by 6 is equal to 1 by 36 is the total probability at on one, right? So you have thrown a 2 dice and the total probability following the product rule is 1 by 36. But what is the probability, total probability, that as you throw the 2 dice, what is the total probability as you throw the two dice and you will be getting the 6 in both the dice? So in that situation, what will be a total probability? 1 by 36, right? And plus 1 upon 36, right? So it will be 2 upon 36, ultimately 1 upon 18. So the total probability in this situation becomes 1 upon 18. So this is but dear students, you will see mostly the application of the product rule in Mendelian principle and rest other principles, because we are going to clarify what is the segregation pattern in law of dominance and what is the independent assortment. So all they together, they are all following strictly a product rule and then addition rule. So let us look at what is the product rule. So law of segregation, when the Mendel performed the monohybrid cross, he gave the law of segregation. Law of segregation. Right? When he performed the monohybrid cross, he gave the law of segregation. What does it say? It says nothing at all. It simply says at the time of gamete formation, half of the gamete carry only one allele, or you can say one copy of a allele goes to one gamete and another copy of allele goes to another gamete. And this is the easiest, but students feel that this is something very complicated process. So let me see you. Okay, what exactly is the law of segregation? Suppose we have the zygote formed over here. This is the zygote, right? And sometimes I would also tell you that Mendel has selected 7 characters, and all the 7 characters were present on all the 7 chromosomes. Okay, though later day of the discoveries proved that some of the characters were present on the chromosome number 6 or chromosome number 5, but still the Mendel was very lucky, or he was very intelligently performed experiment and put forward the idea that all the chromosomes were present on different chromosomes, all the characters were present on different chromosomes. So I will putting you some idea that suppose here is I am giving you the law of segregation, right? What the law of segregation? Suppose we have taken one character into account. Here is a T and here is a T. What do you see? Again, so and this is the what? This is the F1, right? So again there is a female parent. So again this and this. So these are the alleles, right? Now the gamete will form. Again, so simply the law of segregation states that at the time of the gamete formation, now these zygote will form. This organism, F1 is the organism, they will form the gamete. They will form the gamete. So what type of the gamete? One gamete contain only one copy of the gene, another gamete will contain the one copy of the, this is the law of segregation. Means there is also not the law of the purity of the gametes. So see here is the one gamete T having one allele, there is another gamete having a small t, right? And similarly, look here, there is another, here is another, right? So what do you see? You see that the law of segregation, when he conducted the monohybrid cross, then he gave the concept or the law of segregation. He gave the law of segregation. What is law of segregation? It states that at the time of At the time of gamete formation, right? At the time of gamete formation, one gamete carry one copy of gene and another gamete carry another copy of gene, So this is the can you feel it is so easy, but the students generally feel it's very complicated. What is segregating? Actually segregating stands for separation. So what is separating? The genes are separating, which were present in together. The genes capital T, small t, they were present in the zygotic condition. When they form the gametes, they are separating. So the one gamete carrying one allele, another gamete carrying another allele. So this is the law of the segregation. Again, very fantastic thing come to the reality when we talk about the law of independent assortment. When we talk about the independent assortment, but before going through the law of independent assortment, let us use the certain method of representing how to through which you can calculate the phenotype and genotype very easily. Suppose you got the this is the F1, right? This is the F1 parent and this is the F1 parent, and you self this with another similar type of the parent. Then what it makes, half of is the TT, half of is the small t. Here also 1/2 T and 1/2 is the small t. So those were the gametes. So what do you see? Then you conduct what? You do this one, this, then this, then this, then this. Then what do you find? You find 1/4 TT, right? 1/4 TT, right? 1/4 TT. Then you find the 1/4 TT. Is that clear?